Okay, so good afternoon. So what Gang and I, Gang, who I'm not seeing here, so I hope, you are there, sorry, because you're in the dark. Thought to do today is to take advantage of these couple of hours to first to take you through the current status of gene and RNA therapy and gene editing. So I will walk you through the development of this area, which has been really very fast and very impressive in the last say 5 years. And in this context, then we will speak a bit more about AV vectors from the technology point of view, the possible applications, and now of lipid nanoparticles, which are two platforms that we have available here and are very interesting, especially for cardiovascular application. I have been following this field of gene therapy since the early 90s. The first clinical experimentation is in 1990, so 35 years ago. And I'm just back from the last meeting, which finished 2 days ago, of the American Society of Gene and Cell Therapy in New Orleans. So I have updated some of my slides with the latest information. And I'm saying this because I have kept, since a few years, a slide in which I list the application of gene therapy which are approved by the FDA in the United States and EMA. This is around mid-May 2020, mid-May, because this is a period when the American Society of Gene Therapy meeting is held. So this is 2020, so 20 years ago. There were these 7 products which were already approved, the first of which is 2012. This is discontinued now. It was an AV vectors for skeletal muscle, but it has such a low number of potential patients and such a high cost that was discontinued. So let's say these 7 therapies. Then this is the same slide 3 years later, so 2 years ago, 2023. You see that the therapies rose to 18, and this is the same slide now, and there are 25 therapies. So which means that in only 2 years there have been 7 more therapies approved for clinical use. Approved for clinical use doesn't mean clinical trial. It means that you can buy them off the shelves, paying a cost. You can also see here the cost of these therapies, and just to anticipate a theme that we can discuss later, you see that the latest therapies are in the order of 3.2 million per patient, 3.5 million per patient, 3.1, 2.2, which is a huge problem in this area. So basically what I'm going to do is to walk you through this development in technological and also conceptual terms. I was a PhD student several decades ago, and at a certain point there has been 2 enormous breakthroughs in the field of genetics, which was the identification of 2 genes that cause 2 very important and common hereditary disorders. One is the gene for Duchenne muscular dystrophy, and here we are 1987, and this is a gene for cystic fibrosis. That was a massive undertaking because this was before the human genome was discovered, and so people used to take families and restrict through linkage mappings the region of the chromosome where the genes were, and then walk through southern blottings to identify the region that was different. And at that time, it took us, I remember because I was doing sequencing at that time, it took us 1 week to sequence 500, 600 nucleotides. This was a massive undertaking. And I remember that I was asked to give a short seminar on the way the genes were discovered, and I ended my seminar telling that now the genes that are discovered, we know the mutations, in a few years we will have a genetic therapy. Well, now, with 2025, we don't have a gene therapy for that cures Duchenne muscular dystrophy. We don't have a gene therapy that cures cystic fibrosis. This is just to say the difficulty that this area has to encounter. We know now that there are approximately 4,000 genes, the mutation of which unfortunately are compatible with life. The mutation of the other 16,000 genes to make the 20,000 other genome, fortunately are not compatible with life, or are compatible with life only in heterozygosis, so they don't cause a disease, but hereditary disorders are due to mutation of these 4,000 genes. And it was a long time ago that when genes and mutations were started to be appreciated, so in the 70s, 70s are a magic year in molecular biology. It was the decade when monoclonal antibodies were discovered by Stein here in Cambridge, when a group of scientists in Stanford, Paul Berg and others, developed genetic engineering. And this also comes from California. And Theodore Friedman and Richard Rollins said, okay, but if you can transfer a gene into a plasmid and then from a plasmid express a protein and so on, why not transferring a gene to cure a disease that is autosomic recessive? So there the idea of gene therapy started for hereditary disorders. So this is what now we call gene replacement therapy. So basically you have a cell with two non-functional gene copies, so a typical condition of cystic fibrosis. Parents are normal because they're heterozygous. Children inherit one disease copies, and then what you do is you insert a normal copy of the gene. In reality, genes on average in humans have approximately 27,000 nucleotides in length, so they are too big to be transferred. What you transfer is cDNA, so this is not gene therapy, it is formally cDNA therapy. A functional gene copy should be sufficient to cure the disease. This was a very simple-minded idea. It's called gene replacement therapy. Then people immediately realized that we have a problem because DNA has a backbone made by phosphodiester bonds in which the phosphate is negatively charged physiological pH. So it is a huge polyanion that has to interact with a membrane which is hydrophobic and apolar. So never a big polyanion will enter the membrane, cross through the membrane, if this is not facilitated. At the beginning, in the 90s, one of the famous pioneers in the field, Inder Verma, he's one of the founders of the Salk Institute, very, very interesting scientist, one of the most clever I've ever met, was asked by a journalist, Dr. Verma, according to you, which are the main problems that gene therapy has to face to be successful? And he answered, oh, I see three problems, and the problems are delivery, delivery, and delivery. And this remains also now. So the problem of gene therapy or RNA therapy or nucleic acid therapy in general is transferring genes into the cells. However, in those years, there was also here, so we're speaking about the 80s and the 90s, last century, so 50 years ago, there was also another appreciation, that is that there is a class of organisms that do exactly what gene therapy wants to do. These are viruses. Viruses are being selected by evolution to do exactly what we want from therapy. That is to take a nucleic acid, being this an RNA or a DNA, and protect the nucleic acid in the external environment so it is not degraded by nucleases, and transfer this efficiently through the plasma membrane, either directly through the plasma membrane or after endocytosis. After endocytosis, still you have to pass the membrane of the endocytic vesicles. And in particular, in those years, there was a class of vectors that was particularly studied, and these are vectors that belong to the retrovirus families. This was because already in the 20s, there was a scientist, his name was Peter Rouse, who was studying cancer. And he was studying especially a very aggressive cancer in chicken, a sarcoma in chicken. And he discovered that if he takes the sarcoma mass and disgregate it, and then filter it through a filter that blocks all bacteria, then he can take the filtrate, inject a normal chicken, and this chicken in 1 week develops a huge tumor, meaning that there were viruses doing this. And people started studying these viruses, and they ended up with the conclusion that there are viruses that cause cancer virtually in all mammals and birds. So there are viruses they call leukemia in birds, sarcoma in birds, sarcoma in monkeys, leukemia and sarcoma in mice, so name a species. So there was a broad conviction that tumors were caused by viruses also in humans. And people started to look for these viruses in humans, and so there's been a lot of investment, a lot of laboratories working on the classes of viruses that cause tumor in animals. And all these viruses belong to a family of retroviruses. So viruses that are characterized by having an envelope, which is nothing else than the plasma membrane from which they bud, and an RNA inside, which is a messenger RNA, coupled with a capsid, so proteins that protect it. These viruses bind to a cell membrane receptor. They are internalized. And then here, there is an enzyme called reverse transcriptase that converts the mRNA of the virus into the cDNA. The cDNA reaches the nucleus, and there is another enzyme carried in the virion called the integrase that integrates it into the host cell genome. Reverse transcriptase, discovered transcriptase, gave the Nobel Prize in 1972, I think, to David Baltimore and Richard Tainin. And so there was a lot of knowledge, and again, it was perceived. Now you know that viruses don't cause human cancer. There is only one cancer caused by viruses, by the viruses family in humans, which is a rare form of leukemia in the Caribbean or in Japan. That's called ATL, and is due to HTLV1, so one member of the family or the other. But studying these viruses, it was understood that human cancers are due to the mutation of normal cellular genes, which are called proto-oncogenes. They become oncogenes when they are picked up by these viruses. So this life cycle is perfect for gene therapy because you have a cDNA, an RNA that becomes a cDNA that becomes integrated and can provide the gene replacement copy that you need for inheritary disorders. So people picked the simplest virus in the family, which is the Moloni murine leukemia virus, Moloni MLV. And this has only 3 genes, GAR, POL, and ENV, and 2 inverted repeats, 2 extremities that are called long terminal repeats, and asked whether this can be engineered to be a vector for gene delivery. Consider that this virus causes leukemia in mice, and we will come back to this later. This is the virus of Dr. Rouse. So the Rouse sarcoma virus has exactly the same structure, plus has an activated oncogene, the SARC proto-oncogene. This is a normal gene that is mutated in the virus, that is continually expressed. And this is how the first vector were developed. And so basically GAG, POL, and ENV, the 3 genes, completely removed, substituted by a therapeutic gene, and with just some regulatory signals. The long terminal repeat, which is required for reverse transcription and integration, and the packaging signal that tells that this mRNA has to be encapsulated into the virus. So basically, these vectors are produced in this way. There are cell lines that expressed separately in the genome, GAG, POL, and ENV, and you transfer these cell lines with the plasmid that contains the construct I just described. These GAG and POL produce the enzymes that are required for the virus life cycle, so integrase, reverse transcriptase. The M genes produce protein that are on the surface and bind to the receptors, and then this plasmid that you transfect in this packaging cell line generates the mRNA, and then the mRNA has this packaging signal, Psi, recognizes the capsid proteins produced by this GAG gene, and then it's packaged and then gets out of the cell. While it gets out of the cell, emits the envelope protein on the surface of the cell, and so the virion has its surface protein that binds to receptor. That was perfect, but these vectors are very inefficient in vivo. If you inject them in vivo, they are covered by complement, they are inactivated by serum, so very, very inefficient. However, you can use them ex vivo very, very efficiently. And ex vivo means that you have to have a source of primary cells that you can engineer in vivo. And there are three main sources of primary cells that you can obtain. One are cells from the hematopoietic stem cell compartment, so the hematopoietic stem cell that you can recover and keep in vitro, and then, or ex vivo, you say better, and then transduce with the viral vector, and then reimplant. Another one are lymphocytes, you can even expand in vitro, and the third one are keratinocytes, so say in the skin. So people focused on bone marrow samples because bone marrow was a procedure that was already very well established for the treatment of several leukemias. And here the idea was to take the bone marrow, select the compartment of cells that contain the hematopoietic stem cell, so as marked is CD34, keep them in culture for 1 to 2 days, transduced with a retroviral vector with the gene you want to replace, and then re-inject into the patients. And you can think of a number of applications for this kind of procedure. Most of these applications are related to the correction of genetic defect. Let me skip this for a second. And the focus went, for the ease of simplicity and also the medical need, to patients that have, to children that have primary deficiency in the immune system. So the immune system starts, in terms of development, from the hematopoietic stem cell, which then divides, differentiated to a common lymphoid progenitor. And then this lymphoid progenitor divides into two possible branches for development. One is toward the production of a B cell and the plasma cell that produces antibodies. The other branch is toward the production of a common progenitor for NK cells on the one hand, and on T cells on the other, which end up in producing CD4 and CD8 cells. All these come through this route. So if there are defects in any of these steps of maturation, people develop, children develop a severe combined immunodeficiency in which, according to the defect, T cells, B cells, NK cells, or all three or combination don't work. And in the 80s, people had the bad or good idea, you can judge, of trying to grow these children in very separated environment. So basically in plexiglass boxes in which everything that entered the box was sterilized, including toys and food. And these children could grow up until they were almost adolescent. And then at that point, at the very first moment they came out of the void, they were called the bubble boys, came out of the bubble, they got infected and they died quickly. So a very dramatic situation. Obviously, you can imagine there is nothing for this condition. There is an enzyme that is at the top of this development, which is called adenosine deaminase. It is an enzyme that participates in purine metabolism, but it is so it would affect all cells. But these cells, during lymphoid development, there is a very rapid turnover, very rapid replication. So this enzyme is particularly relevant. So it's a cause of immunodeficiency. It's a rare disease. One patient out of 100,000, but a very well-known disease, to a point that there was already at the time a recombinant protein that you could use to somehow mitigate the disease. And in 1990, putting all these things together, so the retroviral vector, the concept of gene therapy through replacement, the adenosine deaminase, the bone marrow transplantations, the first therapy for adenosine deaminase deficiency was performed at the NIH in Bethesda, close to Washington. The first trial in reality was done on lymphocytes directly. Then there have been many trials in hematopoietic stem cell, and this is considered the birth of curative gene therapy. September 1990. There was a lot of hype around, Time magazine covers, lot of books, a lot of hype, but in reality, this was very largely unsuccessful. Very largely unsuccessful because the children survived, but the children, for ethical reasons, were still treated with the recombinant protein, so nobody knew if there was an effect of gene therapy. And what was discovered that if you look at how many cells are detectable in peripheral blood after several years from treatment, they are very, very small fractions, 0.01 to 0.1. There'll be more than 80 clinical trials for different hematopoietic defects. This is the outcome. So basically, what was discovered is that once you take out the hematopoietic stem cell from the body, you keep it in culture for 1-2 days to transduce the retroviral vectors, you need to activate the cells into the cell cycle, because these vectors don't reach the nucleus, these retroviral vectors, gamma retroviral vectors, the right denomination, don't reach the nucleus if the nuclear envelope doesn't break. And so basically you need to activate the cell cycle, but it was discovered that if you activate the cell cycle, these cells also differentiate. And so basically what you correct are the progeny of the hematopoietic stem cell, but you don't provide a long-term cure. Nobody knew what to do until a group in Paris, Alain Fischer is his name, made an experiment which was exactly the same, but for another defect. Instead of adenosine deaminase, he had children with a defect here. So in this protein, it's called the common gamma chain. It's called common gamma chain because it participated in the formation of all these receptors. So the receptor for IL-2, for example, had 3 chains, the gamma chain and 2 specific chains, the beta and the alpha. IL-15 has this common gamma chain plus 2 specific chains, IL-4 gamma chain, 1 specific chain, and so on. And without these cytokines, the immune cells don't develop, and so basically these children don't have NK cells and don't have T cells. And he did exactly the same. So he took the cDNA from this gamma chain, he used a retroviral vector, transduced the bone marrow, but this time this thing worked. This thing worked spectacularly. Here we are in the year 2000. I remember again this American Society Gene Therapy meeting. There were at that time 2,000 people. This year we were 8,000. 2,000 people, a room in which he presented these results. He said, I've treated 11 patients, if I remember well, and 9 of these have exited the bubbles. They go to school, they play in the ground, and they don't get sick. Everybody stood up. There was a big round of applause. The whole community of people, scientists, were completely moved. It was the first demonstration that you can cure hereditary disease. However, these children some of the children started to develop leukemia. There was the first case, and which say, okay, but children develop leukemia commonly, so we don't worry. But then there was a second case of this cohort. All the experimentation for this disease. This disease is called X-KID because this gamma chain is encoded by the X chromosomes, were stopped all over the world, and it was discovered that the cause for leukemia was the integration of this retroviral vector in close proximity to the promoter of the one gene which acts as an oncogene. You remember that the Moloni leukemia virus caused leukemia. And the reason why it caused leukemia is because it keeps replicating, and randomly, after several rounds replication, it can take months, it lands in the promoter region of an oncogene and activates a cellular oncogene. It's called insertional mutagenesis. And this is exactly what happened here. It was observed also in other gene therapy clinical trials. This is a trial for another defect, this time of myeloid cells. So there is insertional activation of all these proto-oncogenes, and so the field was largely stuck because on the one hand, we had the therapy on hematopoietic cells that doesn't work because efficiency is too low, the one for adenosine deaminase. In another therapy, we have a therapy that works because the virus gets in the promoter of genes and activates their replication. So these cells grow up, but then eventually the children develop leukemia. And the problem was solved by a group in Milano, actually, who said, okay, let's concentrate on therapies in which there is not a selective advantage given by the transgene, so adenosine deaminase, and see if we can get rid of the bone marrow before doing the transplantation. The problem was that for ethical reasons, the bone marrow was engineered in vitro, but then was injected with all the rest of the bone marrow of the patient intact. So basically, what these people did was to say, okay, let's get rid of the bone marrow. There is a drug that's called busulfan, which is a drug that is used for chemotherapy, that has a very dose-dependent toxicity. So the more drug you give, the more you kill cells. And so they gave a suboptimal drug dose to kill the vast majority of cells, but not all of them. So in case something goes wrong, there are enough cells to repopulate the blood. And at that point, they did transplantation, and this was the real first therapy that worked. And so they could cure adenosine deaminase deficiency with this non-myeloablative conditioning. So we are 2002. And this therapy has has had a strange future because the therapy continued to work. This is an update 2009. This is an update which is even more recent. Survival 100%, 80 patients treated. All these patients are perfectly normal with no sign of cancer, no sign of immunodeficiency. They go to school, they live normal life. They are now young adults with no problems, but the therapy was licensed to a company here in the UK from Milano, which is Orchard Therapeutics. And Orchard Therapeutics ran into financial troubles, and they had to suspend the therapy because this is a rare disease, so 1 people out of 100,000. And so they couldn't make sufficient revenues. And so they stopped the development. So there is a therapy that works beautifully, that is proven, but there are no financial means. Now a charity in Italy, Fondazione Teleton, has taken the rights and they are redeveloping the therapy in Italy. This also gives you immediately the idea that something is medicine, something is research, and something is business. But if you don't have all these 3 components aligning, it's very difficult that you end up anywhere. In any case, that was a big success. The problem of insertional mutagenesis was solved in another way by moving away from the monoclonal leukemia virus to HIV. HIV is a non-oncogenic virus. It causes immunodeficiency. It is a member of the family that is a bit more complicated. It has these GAG, POL, and DEL genes, but in addition, it has 6 regulatory genes, which are these here. And an Italian scientist now, who's also in Milano now, his name is Luigi Naldini, was a postdoc in this Verma laboratory at Salk, developed the first lentiviral vector. So they are called lentiviral vectors and not HIV vectors at the beginning because not to scare patients that they wanted to cure their children using HIV. At that time there was not immunotherapy that was successful with HIV, 1996. And these vectors don't integrate into promoters. They have a much broader integration capacity. They are not, they don't do insertional mutagenesis in the sense of causing cancer. And so the field has skewed away, shifted away from lentiviral vectors to the use, from the retroviral vectors to the use of lentiviral vectors. These are two examples from the work on Aldini, again in Italy, of therapies for children for two other genetic disorders, metachromatic leukodystrophy and Wiskott-Aldrich's syndrome. So now, if you look at now the 25 therapies that are approved, there are 3 therapies based on these gamma retroviral vectors and 9 therapies with lentiviral vector. You see that all the most recent therapies, so here we are from 2022 to today, you see whether all this was up to 2017. And here it is from 2019 to now, and also just from 2022, all these therapies have been approved. All the most recent therapies are based on these lentiviral vectors, and there are 15 hereditary disorders that are cured by gene therapy, and for which one can find therapies. They are listed. They are listed here. Okay, let me skip this. Now, if you look at the overall clinical trials that have been performed on the gene therapy since the beginning to now, there are more than 3,900. So this means that these are trials in which patients have been injected either ex vivo on their primary cells, infused, or in vivo with vectors. But you immediately see that the vast majority of trials are not for monogenic disorders, but are for cancer. Monogenic disorders are less than 10% of the population affected, while cancer kills 25% of the population. Cardiovascular disorders kills 30% of the population. So since there is a huge company involvement, companies want to do something that is very profitable. And so most of the attention was for cancer. And nothing worked at the beginning, nothing worked. Except when the attempt has been at using gene therapy not to target cancer cells, but to target the immune system. And all this started from work that was performed again in the 90s by a group at the NIH. Steven Rosenberg is now quite old, but still very active because he has his clinical practice. I don't know if he operates anymore. He was a surgeon in any case, an oncologic surgeon. And he observed that if you take cancers that he removed from patients, he was working with melanoma in particular, there were a lot of lymphocytes infiltrating this cancer, which was somehow an indication that the immune system tried to fight cancer, to kill cancer cells, but it is relatively inefficient. So he developed a method to recover these lymphocytes from a cancer sample after the operation of the patient, or even from biopsy of the cancer, and expand these cells in culture. You know that T cells are one of the few cell types that we can expand very easily in culture because they respond very well to a cytokine, which is interleukin-2. And so basically he expanded these cells and reimplanted these cells into patients. And he started seeing that there is an anti-cancer response. So he called this tumor infiltrating lymphocytes. So they are called are named as TILs. 2 years ago, there is also a product, a cell product based on TILs that is also being approved by the FDA, is available in the United States. So basically, the idea is that it is lymphocytes that must be our target to cure cancer. And so based on this, people say, okay, but if I have to take lymphocytes from the cancer itself and expand them, how many of them can I obtain, and how efficient the procedure, how long does it take? It's a very difficult proposition. And so someone started saying, okay, but why don't we take any lymphocytes, but we redirect lymphocytes against a given antigen? Again, in the beginning, there was a lot of skepticism on this, it will never work, so on and so forth. But to make the story short, this was the birth of the CAR-T technology. So basically, lymphocytes, in particular CD8 lymphocytes, so cytotoxic lymphocytes, recognize their targets through a complex of proteins that's called the T-cell receptor, which is anchored to a signaling part, which is called the CD3 antigen. And so basically the T-cell receptor is an alpha and a beta chain, recognizes the antigen. And these are other proteins that are help transferring the activation signal, and all ends up in a kinase they call ZAP70. So basically, people started to develop synthetic molecules in which the recognition of the antigen is by a modified ligand binding domain of an antibody. So basically the variable light and variable heavy chain of an antibody, again, which is linked to the signal transduction component of this Z chain, which is bound to, which recognizes ZAP70. So basically this signal transduction arrives at ZAP70 but through an antibody. This was called the chimeric antigen receptor, CAR, or T cells. And to make the story short, this was really one of the most successful therapy. Here the application is patients. From the patients, one collect his peripheral blood cells, and then in the laboratory these are transduced with a lentiviral vector that expresses the CAR-T constructs, and the CAR-T construct has a specific antigen, and so basically one obtains millions of CAR-T cells which are cultured in the laboratory, reinfused in the patients. And the first results were obtained in 2017 and were really spectacular. The antigen, surprise, surprise, however, is not an antigen specific for tumors, but the target is a normal antigen expressed on B cells, CD19, but this is a perfect antigen for specific cancers of the lymphoid system, which are B-cell leukemia and B-cell lymphomas, which are due to the expansion of B cells where this protein is highly expressed. The results were spectacular. The first, this is an editorial, the first trials were performed in patients who had B-cell lymphoma and B-cell leukemia, were refractory to any other therapies, so they were destined to death. And the success rate was up to almost 90% in one of these trials. So these patients were from dying to cured completely and permanently. One enormous success which paved the way to the development of CAR-T. The first two products are 2007 from Novartis and Gilead, and then progressively there are now 7 CAR-Ts which are approved. The antigen, however, are always the same, so they are CD19, and there is only one new antigen, which is a protein that is expressed in multiple myeloma. And so basically all the conditions for which we have these CAR-T therapies are cancers of B cells, so acute lymphocytic leukemia, B cells or B-cell lymphoma, or multiple myeloma. The whole field of people working on CAR-T is now desperately trying to find ways of CAR-Ts working on solid tumors. But this is an interesting category that has been really very successful. Let's not forget that there is a possibility of using CAR-T also for cardiovascular application. I'm picking up one of the first example in this case. This is a work from Jonathan Epstein from Philadelphia, who developed CAR-Ts against an antigen that is expressed by activated fibroblasts after myocardial infarction, so the FAP antigen. And he shows that if he does CAR-Ts that kills activated fibroblasts, he reduces fibrosis and improves myocardial infarction. Working with viruses, just for the sake of completeness, this is very fascinating. There is also the possibility of using viruses that replicate in the body. This comes from very fascinating stories from the 18th centuries, in the medical literature, there are cases of patients with very big tumors in very sick conditions. So they call the priest, they give all what has been done, the patient is unconscious, then suddenly he gets a very high fever. So he got infected with something, very high fever, the whole family waits for the patient to die, but he survives, stays better and better, he recovers from the infection, and the cancer is gone. There are a few anecdotic cases which led to the idea that there might be some viruses that selectively replicate in cancer cells. And this was taken by a group of people in Canada at the beginning, at McMaster University. And basically what they thought was to do these viruses made on purpose, and they focused on adenovirus. Adenovirus is a virus that is very effective at exploiting cells for its own replication. When it enters the cells, he has a protein called U1A that blocks, that basically hijacks all the cellular function or its own replication. The cell recognizes there is double-stranded DNA and reacts by activating p53, but there is another protein from the virus, which is called U1B, that blocks p53. So the virus can replicate abundantly and kills the cells. So you can develop adenoviral vectors. They are very inflammatory, but you can develop them by knocking down both U1A and U1B. So basically, you don't produce the virus. But if you produce a virus that has only U1A, so it replicates very well, but doesn't have U1B, this virus activates p53, so it doesn't replicate. However, more than 50% of cancers have p53 deleted. So basically, if you give this virus to both normal cells, the virus don't replicate, and cancer cells don't have p53, so the virus replicates and kills the cells. And there's been a lot of experimentations for this until one of these viruses reached the clinical application. It is a complex adenoviral vectors, which is also, as I say, hard. So there's also inside the genes for some cytokines, and it was approved for clinical use in 2015, and which is this one is called TVEC for melanoma. There is another one that was just approved 2 years ago, which is instead a herpes simplex virus, which is made in a cream. And there is also an adenoviral vector which is used for bladder cancer, so in patients with cancer of the bladder, to express interferon alpha beta. You inject it into the bladder. All these vectors work basically, especially retroviral, antiviral vector, work ex vivo. No? So you need to have lymphocytes, you need to have hematopoietic stem cells, but what do you do with the diseases in which you have to transfer the genes in vivo? And here there is another vector, and this vector is based on a virus that's called AAV. I will tell you better of AAV after I finish this story on gene therapy. For now it is sufficient to say that it is a very small vector, about 20 nanometers. There's only 2 genes, rep and cap. You can remove completely the 2 genes and put the genes you want, a promoter and a polymorphisation site, if it is a cDNA. And the only part of the vector that works are 2 inverted terminal repeats, so 2 hairpins with this structure. Now, if you want to develop an AAV vector and transduce your cell in the incubator, it is the worst vector that you can use. It works very, very poorly for all the cells that replicate. I will tell you something more why probably this is the case. However, if this vector is injected in vivo, it works very well in the retina, so both the ganglion cells, so the neurons, or the photoreceptors, the neurons, again, if you inject it in the brain, the skeletal muscle fibers, the heart. These are all from applications that we did over the years. So it works in post-mitotic cells. So cells that have exited the cell cycle and will never re-enter the cell cycle again. But for example, I mean, endothelial cells also don't replicate. In normal conditions, endothelial cells replicate every 6 months, but they are not post-mitotic because they can respond to a stimulus. And in fact, AV is very bad in these cells. While cardiomyocytes will never re-enter the cell cycle unless stimulated, I should say. And so it works very well. A neuron will never re-enter the cell cycle, so it works very well. Very interesting biology. One of the organs in which there are no replicating cells is the eye, and you can this is the cornea, this is the optical nerve. You can inject a vector from subretinal, so you detach the retina transiently, and so this reaches directly the photoreceptor, the retinal pigment epithelium cells, or you can inject in the vitreous. In this case, it meets first the neurons, so the ganglion cells, so the one that ends up in the optical nerve. Now you know that the structure of the retina is inverted from what we expect. So the light has to go through the neurons to reach the photoreceptor. These are some examples from our past work. So this is injection in the vitreous. So you see this is an BHF, and you see the ganglion cells, but not the photoreceptor. If you inject the subretinal, you see all the photoreceptors, the retinal pigment epithelium cells that are transduced. There are hereditary conditions which are autosomal recessive that can be cured. The eye is perfect for gene therapy because it is a restricted environment. It is immunologically separated. You need a very limited amount of vector. You can inject exactly where you want. And there are defects in the what's called the visual cycle, which is the cycle of regeneration of retinol after it has been hit by photons, which this cycle is all carried out by enzymes. If you lack some of these enzymes, then basically children are blind. One of these diseases is a deficiency of RPS 65, which makes children blind at birth. The mother realizes the children are blind because immediately after birth, the mother, the vision is very blurred in the newborn, but the mother, the newborn has a fixed focus length, which is more or less the distance from his eyes and the eyes of the mother. So the mother immediately, from the first days, understand that she can get an eye contact with the children. These children are blind, so basically this eye contact is lost, and they're blind forever. So there's nothing you can do. There is an animal model for this defect of this RPE65 enzyme, which is a dog, which is spontaneously blind, so it's not genetically engineered, and a group in Florida developed an AV vector expressing a normal copy of this RPE65 gene. Again here, meeting of the American Society of Gene Therapy, room full of people, 4,000 people at the time, so we are some years later, and they show a movie in which there is one blind dog, and they put the dog in one corner and food in the opposite corner. And in the middle, they put a lot of obstacles. They type these, how do you call these cones on the road work? Cones, okay, all these cones. So the dog smells the food and rushes toward the food, but he hits against all the obstacles he has, so he really doesn't see. And then they inject in one eye the AV vector, and after a couple of months, they repeat the same experiment with the same dog. They put it in one corner, and the dog goes toward the food, but at this time, it avoids all the obstacles, and he runs using in front the eye that was corrected and not the other one. So this was really, again, a moving moment in history of gene therapy. And clinical trials start. This has been the first successful AV vector that was developed with the name Luxturna by a biotech company in the US called Spark Therapeutics. And the problem is that children regain vision, but regain vision only if you do the therapy very, very early in life. Because if you do later, then the central nervous system and the eye itself is not able to adapt to the new signals that it receives from the retina. So it is still a problem that is only partially solved. However, again, in one of these meetings, to make a show, they took a child who, which they showed the movie, it was really almost blind. Before had to walk with a stick and so on. And then while the presenter was giving her talk, he asked the child to sit at a table, and he was writing his homework for school while he was speaking, showing that he was really reading and writing. And so it is an amazing advancement. Hemophilia B is another target. Another game changer was an AAV vector expressing a gene to cure spinal muscular atrophy. This is a disease in which you can overcome by overexpressing a normal copy of a gene that is mutated, and it has been really very, very successful. These are children. There are different stages of the disease, different categories, but the worst cases, children are destined to die in the first years after life, and these children now walk and they survive and they move. Really game changing. And these are the 8 AAV applications that you have. The latest one are the one that not particularly effective. Luxturna is somewhere here. Here it is. It was one of the first. The latest one a bit less effective. This is one with a microform of dystrophin. Dystrophin works by anchoring the DGC complex, so dystrophin glycoprotein complex, to actin. And if you have mutations that destabilize, that disrupt the C-terminus of the protein, then you don't have this anchorage, and this is very bad. However, if you have mutation in the middle, but the C-terminus and N-terminus are still there, then the disease is converted to a very milder form, which is called Becker muscular dystrophy. So basically, these mini dystrophins are dystrophins that are sufficiently short to fit into an AAV vector, but it doesn't work that much. It was anyhow approved last year. This year, there's been the approval of, always last year, there's been an approval of factor 9, expressing the cDNA for, an AAV vector expressing factor 9 for hemophilia B. But here it seems that patients don't want to take it because the substitutive therapy with recombinant factor 9 is so effective that people are a bit scared of getting a gene therapy surrogate. So if there is a disease for which there is no cure, something, but if there is a disease in which there is a conventional cure, then the perception is different. Then gene editing came on. Gene editing is basically based on a nuclease that cuts DNA, and the normal way of cutting DNA, of repairing this double-strand cut, is through non-homologous enjoining, and which is carried out by the cell. So basically, gene editing is not a form of repair, because repair is by the cell, but it's a form of cutting. And people started desperately to find ways of cutting in close proximity of mutation. The interest is enormous, because here you could target autosomal dominant mutations. So in autosomal dominant diseases, you cannot add one copy of the gene, but you have to delete the mutated copy. And so started to develop 2 types of protein-based nucleases, which are sequence-specific, zinc finger nucleases, TALE nucleases. Very difficult proposition. So basically, you have specific amino acidic modules that each one recognize one base, and you have to put them together. There was a leading company called Sangamo who has a patent of this. They arrived to a clinical trial for HIV to kill to destroy one of the receptors of HIV. This is the first gene editing patient that reach gene editing therapy that reach patients with the same principle for mucopolysaccharidosis, but efficiency was by far too low. So these are all was abandoned because in the meantime there's been the discovery of the CRISPR-Cas9 system, which is a system that was developed, was discovered in bacteria, is a sort of immune system of bacteria, and these two investigators, Jennifer Doudna and Emmanuel Charpentier, discovered that you can minimize the system to only two components. So an RNA that guides an endonuclease to a specific site. You can transfer this to any cell and this cleaves DNA. They won the Nobel Prize a few years ago. From that point, there's been these crazy applications to all types of conditions for discovery, to create genetically modified animals. I don't enter into this. Very difficult to apply to humans, but there are some conditions in which you can do it. For example, again on dystrophin. No? So if you have, for example, a mutation of this of dystrophin in exon 50. If you have a mutation, this exon doesn't exist. The splicing machinery in patients brings together exon 49 to exon 51. The problem is that exon 49 and 51 are two different open reading frames. So basically the protein that comes out is truncated at 59, then there is a stop codon. And so dystrophin lacks this anchoring property. Now, if you, instead of having only the deletion, you also induce the deletion of exon 51, the cell then splices 49 to 52. And so you lose two exons, but you join, you have a protein that is on the open reading frame. So you have both the C terminus as the N terminus. And this is one of the clinical trials that are currently ongoing. And so the idea is to take CRISPR-Cas9, target this to avoid joining of this exon, and so cure the disease. There are some applications that are more advanced in clinical trial. One has been already approved, which is this one. And it is for beta thalassemia and sickle cell disease. So these are diseases due to defects of the beta globin, so the beta chain of hemoglobin. And basically it is known that the adult hemoglobin, so including the use of the beta globin, starts only at birth, while in the fetal life there is a gamma chain that has more affinity with oxygen. That is the prevalent form. There are some individuals in which this doesn't shut off and keeps being expressed throughout life. So these are people with persistent of this fetal hemoglobin. And so the idea here is to use CRISPR-Cas9 knockout, so to cleave in proximity of a silencer regulatory element that blocks expression of the gamma chain. So basically to reconstitute gamma chain expression in patients with the beta thalassemia. And this has been the first gene editing clinical application that was approved by the FDA it was in December of 2023. There was a big meeting here in London in January, in which they called to speak to the podium a patient with sickle cell disease. Sickle cell disease is a mutation of beta globin, in which a single point mutation in which you have the hemoglobin precipitated in red blood cells. So these cells pile up and clog the circulation. So this patient has tremendous pain because they have microinfarcts throughout their body. And this lady came to the podium and said, listen, my life was absolutely impossible. I couldn't attend anything. It was a disaster. I was miserable, and so on. they did this gene editing intervention, reactivated fetal hemoglobin, and here I am, I'm moving, I'm speaking, I'm traveling, I'm living with a family and so on. Again, big round of applause and so on. The problem is that this costs more than 3 million per intervention, considering that sickle cell disease and beta thalassemia affect mostly developing countries. There are more than 2 million people only in Africa with the disease, I mean, who can afford 3 million per person. So huge problems. Good. Going toward the end, this is also interesting. This is a gene editing application that is raising a lot of ethical debate. I'm totally disregard this. So basically here the idea is patients with hypercholesterolemia. Patients with hypercholesterolemia have a huge benefit at inhibiting an enzyme, which is a normal enzyme, it's called PCSK9. This enzyme is involved in cholesterol metabolism and in recycling of the receptor. So if you knock down this enzyme, or block this enzyme, you have more recirculation of cholesterol, and so you lower the cholesterol levels. And this company, Verve, wants to knock out this receptor, this PCSK9 patient, so permanently patient. So is it ethically approvable, a therapy that targets a normal cellular gene to try to cure a disease risk factor, not the cause of the disease? So in the case of hemoglobin, you somehow cure a disease. Here you simply cure a preventive factor, so the levels of cholesterol. However, they are having a lot of room, especially in the United States, because the market, you can imagine, is absolutely enormous. And well, I think I can stop here. Questions?
What are the steps in the process of producing and licensing a drug which lead to the really high costs? Which are the limiting factors?
So if you ask this question to a biotech person, they will tell you, we had to have an enormous investment, clinical trial costed us no less than 30,000 pounds or dollars, and the cost of producing, for example, an AAV vector is astronomical because we have a regulatory constraints and so on. The part of the reality is also these people in companies have really a different value of money compared to also in terms of salaries compared to people in the academia. But the reality is that the costs are not calculated on the real cost of the production, including also the amortization of the expenses for the development, but they are calculated on how much the society would save from standard cures for these patients. For example, if you have a patient with sickle cell anemia or beta thalassemia, he needs, apart from his disease that is very, very painful, very problematic, and he has a shortened lifespan and so on forth, but he needs continuous transfusion. So how much is the cost of the transfusion over a lifetime? Less than if you spend 3 million to cure him, definitely. But this is essentially wrong. It is a market-driven calculation, which really has to change. There is a lot of debate on this. But consider that, for example, this therapy for beta thalassemia, not based on gene editing. We have two therapies for beta thalassemia. This one based on gene editing, another one with the cDNA for the beta globin, was developed by a company called Bluebird Bio. And they costed it $3-something million. They came to Europe, they started speaking with regulatory authorities in Europe, started speaking in Germany. Germany said, oh, no, we are not going to pay for this with the national health system. and none of our patients can afford this amount of money. They went to France, the same, Italy, the same, European Community country, the same, UK, the same, and then they had the regulatory approval in Europe, and they abandoned it. They went only to the United States, thinking that the situation is more favorable because there are private insurance companies and so on and so forth. I was told that they are on the verge of bankruptcy now because nobody can afford these prices. So, but this financial issue is really problematic. CAR-Ts is a bit different because the number of patients that can afford the therapy is much that are in need of the therapy is much higher, and so the cost of a CAR-T is now reduced to $200,000, $300,000 per patient, which is much more affordable also for the insurance companies. The new trend is then not even do the lentiviral vector transduction, expansion of cells, and reinfusion to patients, but develop lipid nanoparticles targeted to T cells with an antibody, and they do the intervention after you inject them systemically. And there's a huge interest in doing these targeted therapies with nanoparticles directly in vivo.
Presumably, the list that you had updated, those are all protected with patents. So
multiple.
Right, but then there might come a time where
yes,
those patents will run dry.
Yeah.
Maybe new technologies will start to surface. But in terms of those patents, once they run dry, do you think that there's a chance that these can then be passed on to developing countries, like you were saying before, where there's a need for beta thalassemia in Africa, or where
it could be, but these patents will with patents and all their extensions will come off in 20 years, 25 years. So, and as you said, technologies will change. If we had to bet, for example, you have all these therapies for hereditary disorders, factor 9, factor A, AADC, arysulfatase, SMN1, beta-globin. All these will not be there in 10 years because they will be substituted by gene editing. So gene therapy through replacement is something that we see in this window of history of medicine. But the future, it will be all gene editing, all genetic corrections, and then for several of these therapies is not only the cost of the therapy, but you need also the right environment. So so CAR-T, for example, you need a laboratory that can expand the cells and the clinic that can give the cells. So all of these are not necessarily available in developing countries. Yeah. So it's more a matter also of administering the therapies, not only having it available. Consider that if you think something much simpler, which is recombinant protein production. I've not followed this recently, but until I was at ICGV, which was the center of the United Nations working with developing countries, in the whole Africa, there was not a single country that could develop any generic for recombinant proteins, insulin, about the antibodies, trastuzumab. So the simple things that are way of patent, they don't have the capacity, the human capacity to produce it. The human capacity is a bad center. The ecosystem, yes, the infrastructure. Oh. And so these are advanced therapies that are very difficult to transfer to developing countries, which increases even more the divide between the third world and the first world.
If you want to do gene editing in vivo,
you like the idea, no? I knew that you would like it. Yes.
It seems in terms of licensing a drug, maybe not for kind of emergency and experimental approvals, but for licensing a drug, it seems that it would be important to have very detailed studies where you have deep sequencing of patients who've been treated, these sorts of things.
Well, what do you mean with the term licensing?
I mean approval for market.
Okay.
Like FDA approval.
Okay.
Do you think it's problematic that if you're doing a gene editing intervention in a patient, hopefully at a young age, potentially you can't get a sample from that patient for decades before you can properly analyze it to understand whether you have off-target effects or these kinds of things? Is this a problem or?
But not necessarily. So you do what you can, and you have always to balance it with the risk posed by the disease. If you have a disease in a child that brings this child to death, and you can run some risks of seeing a cancer after 20 years.
So if you see an improvement in terms of outcome, you think it's enough to? No.
The regulatory authorities have a set of rules which you have to prove safety. The first experimentation, the phase 1 experimentation are not even for efficacy; they are only for safety. And part of the safety package is, for example, to see how many, speaking of gene editing, how many off-target effects you have in other organs that can be hit by your drugs, and so on and so forth. So you have to do these kind of studies. You have to do these studies in large animals, in small animals, but by definition you cannot be exhaustive on this, and then you have to balance with the risk-benefit balance.
This is why I mean, just in human patients, it's very difficult to assess the effects on different organs when the patient is alive.
Yes, so you do it in mice, you do it in pigs, you do whatever you can in humans, for example, peripheral blood, and then you save the patients. And if in 20 years they will develop a leukemia, they will develop leukemia. But at least they have lived. Okay, if you bear with me 15 minutes more, I tell you something about AV vector specifically, and then I leave the floor to Gang.
Can we have a quick break?
Oh, yes, yes.
Like 5 minutes.
Yes, 5 minutes.
Do you want me connect or I give you my slides?
Give me your slides.
Okay, it's in here.
But I don't have this.
I can do AirDrop.
I have only the.
I have the connection.
While I have you for 5 minutes, do you think there is a possibility of us trying to use the AV Maya for my project?
Yes, but you don't have
plasmids today.
You don't have an advantage.
Why?
Because it doesn't work much better than AV9.
It doesn't work.
No.
But I thought it would.
Sorry, it's not here. That's this. Oh, no, the other side.
No, it doesn't work much better.
I thought it wasn't.
Yes.
No.
It's better than AV6.
It depends. If you inject intracardiac, probably AV6 is even better.
But if you inject systemically?
It is more or less like AV9. It can be a bit better, but it is not tenfold better. It can be twofold better.
But twofold better in my context is important.
I would try with an AVGFP before embarking in redoing vectors.
I mean, yeah, okay, we can discuss that.
Yeah, who is giving you the?
He's mentioned it to me. That was, I don't know.
Maybe they're giving you the capsid.
No, she said that there was some collaboration or somebody had a capsid.
Yes, no, but the capsid, so someone is giving you the vector or is giving you the capsid to produce the vector?
No, the capsid. The vector I have myself.
The capsid. And so you need someone that produces the vector.
I don't know, maybe I need to speak to example, but she explained to me that it was possible.
Yes, but this is, we have to send to Trieste and ask Trieste to produce the vector. Yes, yeah, we can do that.
This is what I mean.
You can do, but before embarking with your construct, I will do an BHF, and so we see if it is really better. Because this is what I'm saying. So if you have a capsid, you produce a vector with GFP, that capsid. You compare with the V9, and so we answer this question once forever.
My vector has a GFP in it, so I can do both.
Has a GFP inside.
So I can do both at the same time.
Okay, then you can do.
Okay, thank you.
But don't expect wonders.
No.
The best demonstration of what I'm saying is that if you look around, people keep using AV9.
But AV9 is new, no?
Now it's 3-4 years. Even more. It was developed by Dear Grimm, no?
I need to read more.
Well, there is a question online. So I noticed approval is for FDA and EMA. What is the status for MHRA approval in the UK for these gene therapies?
I have no idea. I don't think there is a separate. There are some products that are separately approved in Japan, which are not in the list, or in China, several of them. But I don't believe there is. No, I'm sure there is not a separate something that is approved by MHRA and not by EMA. And the gene editing was approved first by MHRA a couple of months before the EMA. So I would tend to consider that what is EMA is also MHRA. Also because companies don't have any interest in having something licensed in the UK specifically without the European market or the European and US market. Okay, a few words on AAV. 1960, as you understand, I like seeing the development of science in chronological terms because it tells you something about what happens and also on people who did this. There is a group of researchers who are culturing cells infected with adenovirus. And they realize that these cells also contain a virus that is much smaller than adenovirus. And they call it adeno-associated virus. It is much smaller, has nothing to do with adenovirus. This is a simple reason is that this is an electromicrograph. The simple reason is that this is a defective virus, so it's not capable of replicating on its own, and it needs co-infection with another virus, in this case adenovirus, to be able to replicate. The virus is much smaller. This is adenovirus. This is AAV. It is a member of the family of the parvoviruses. Parvo in Latin or Greek? Parvos, Latin, means small. And it has only 20 nanometers. Just to give you a reference, HIV is 120 nanometers in diameters. The nanoparticles that gang produces are 100 nanometers. SARS-CoV-2 is 110 nanometers. So this is 5 times smaller. So really very small. And it has a single-stranded genome, which is also very unusual. And the single-stranded genome can be one strand or the complementary strand. So it can be one polarity or the other polarity. Usually it is 50% of each. So far, parvoviruses, there is a famous parvovirus, we call parvovirus B19, which is the causal agent of some diseases in children. But this belongs to another family, the family of the helper-dependent parvoviruses, to which AAV belongs. Has never been associated to any disease. And so it is non-pathogenic, but all of us have antibody against it, 90-something% of us have antibodies. And we acquired this antibody during our infancy. So it's a virus that circulates in the population. And so we are all in contact with them, but without causing the disease. And the virus is very simple genetically. It has only 2 genes. REP, which encodes for all the replicative functions of the virus, we go to it in a moment, and CAP from the factors that surrounds the DNA, which is single-stranded with these 2 herpes. The herpes has approximately 150 nucleotides. So basically, a virion is basically formed by 60 repetition of a protein coming from CAP that surrounds a single DNA. Very simple, super simple. And you can produce the vectors by removing completely REP and CAP. So all what remains are these 150 nucleotides, and inside you can put whatever you want. Molecular biology term, the situation is slightly bit complex because there are 4 different REP isoforms, which are formed by using 2 different polydelegation sites and 2 different transcriptional sites. So there are 2 promoters, but basically they are all come from the same gene, and 3 different capsid genes, which use a different AUG starting site. So they're called VP1, VP2, VP3 from the same cap gene. In addition, more recently, there have been discovered 2 very small accessory proteins, not very clear what they do. They're called AEP and MAP from 2 operating frames in this region. This is a structure of the inverted terminal repeat. This is required for replication, and replication is thought to require the 3' end. You know that all polymerases in every living organism on the planet always move in the 5' to 3' direction. There's no polymerase that does the opposite. So they stand 3' OH. So basically the way it replicates is a polymerase, which is a host polymerase, can be DNA polymerase alpha or one of the repair polymerases that extend the 3' OH and copy the template until the end. And the virus hits a cell, and then if the cell is infected by adenovirus, but also by herpesvirus or papillomavirus, then it replicates. It forms many viral genomes, half plus polarity, half minus polarity, gets encapsulated and lies the cell. If the cell is not infected, then these helper functions are missing, and the virus gets double-stranded through a DNA polymerase. It integrates site-specifically in a region of human chromosome 19, chromosome 19, Q13.3. Only example of site-specific integration by a virus known. These helper viruses don't code themselves something that helps replication, that is required for replication, but generate a stress to the cell by which it is the cell that does something that by which it becomes permissive to replication. It is a cell, and so replication can be also induced by physical agents, which are very broadly variegated: gamma radiation, X radiation, UV radiation, heat shock, or a series of chemical agents, methylmethanesulfonane, metomSSC, cisplatin, hydroxyurea, topoisomerase, and so on. Come back to this later. There are 9 natural serotypes known. These are serotype in virology is defined from a variation in the surface protein of the virus by which it is not recognized by sera of animals that have been exposed to another virus of the same family. So there are 9 different serotypes from 1 to 9, and they all have different receptors. These receptors are usually very broad receptors on the cells. And so, for example, AV1 binds to N-linked sialic acid. This binds to heparin sulfate proteoglycans 2 and 3. This to O-linked sialic acid, N-linked sialic acid. So, I mean, proteins that are present on the surface of most cells. However, despite these surface receptors are quite spread, either for the presence of some secondary receptor that help entry, so for example, FGF receptor or hepatocytofactor receptor for AV2, platelet derived factor receptor for AV5, EGR receptor AV5. These are co-receptors that help in the realization. Either because of the presence of these, or mostly for something that occurs inside the cells, some serotypes have a different specificity. For example, AV9 is the one that in mouse is the best to transduce the liver, or AV2, sorry, to transduce the heart, or AV6 goes well also to the heart, but also to the liver. AV8 goes to the liver and the pancreas, and so on. In addition to these 9 serotypes, there are a vast number of artificial serotypes that have been developed over the years by different techniques. So these are the natural serotypes in humans. Then there are natural serotypes in monkeys. Then there are specific mutations introduced in the capsid proteins, or people took the capsid protein on one serotype and fused, so did the chimera with part of the capsid protein of another serotype, or did the capsid shuffling. So you take all the serotypes, you break the genomes, and you use PCR to make them recombine. So you obtain a library of different variations. That is called capsid shuffling. Or people inserted small peptides in 1 or 2 regions of the capsids, peptide libraries, and selected for variations that are specific for one organ and the other organ. So we have a myriad of these, several of these artificial serotypes, not because only because it is interesting to understand the serotype, but because if you are a company, you want to develop your own, not to depend on the license of someone else. This is the reason why people, and then everyone says, oh, no, mine is better than the other, and so on. In reality, I mean, the natural serotype cover most of the applications. So the vector is made like this, so therapeutic genes, and so I say transcriptional cassette, and then the 2 hairpins, and the production is by transfecting this plasmid into a cell, which is also co-transfected with a helper plasmid. This helper plasmid contains the rep in cap gene, but without the ITRs, and these genes of adenovirus that induce the cells to become permissive for replication. In these conditions, the rep cleaves out the vector from the plasmid here, start replicating it, and the cell produces the capsid protein, and then the virus gets packaged, and when there are 10 to the 5, 10 to the 6 viral particles produced per cell, the cell lyses, and it releases the vector in the supernatant. You can use these vectors to obviously express cDNA with the POL2 promoter. We use mostly either the CMV promoter or the troponin I promoter if you want to have cardiac specificity. Or you can also express a short helping RNA. So basically you use, in this case, a POL3 promoter. We use the U6 promoter, and then a helping RNA for a siRNA generation. We have set up a facility in the early 2000s, so 20 years ago, in Trieste, together with Lorena Zendini. They still produces our vector for us until the end of this year, and in 2004. And basically, this facility produces approximately 250 batches per year, so large number of vectors produced, and we produce basically all the serotypes. This is not even an updated slide. And there are different ways of purifying these vectors from the lysate. A way that is not most effective for scaling up, but is very effective for, let's say, academic use, is to run the vector through a cesium chloride gradient. And when you run the vector through a cesium chloride gradient, the vector bonds in a certain position, and it bonds by forming two bands. One contains mostly empty capsids, which you don't want in your preparation because it can compete for the entry into the cells. And one contains the full capsid. And so basically you recover this fraction here. If you want to inject small animals, this is what we do. We do dialysis of this and then we use it. If we want to inject a large animal, we take this and run a second cesium chloride gradient. This is very ineffective if you want to produce very large quantities for, let's say, GMP production, clinical trial, whatever. And so companies, instead of this simple procedure, they use chromatography. So they put the vector on top of chromatographic columns, and then they recover a different fraction. They have more problems at recovering the distinguishing the empty from the full capsid, but there are now very, very established protocols for this. As I said before, the vector is very effective for postmitotic cells. If you take an A9 and inject intramyocardial, then you transduce well the left ventricle. These are 2 injections of 30 microliters in the left ventricle. This is an AVGFP with the CMV promoter in adult mice. If you inject systemically, in this case it is retroorbital, but it's not very different. It's a bit more efficient than intravenous. It goes into the circulation and transduces the whole right ventricle and left ventricle, a bit less efficient, but more homogeneous.
Sorry, Mauro, can I ask about the retroorbital injection?
Yes.
Is that something you did in Italy or?
Yes, no, in Italy.
Okay.
I would be in jail.
Okay.
I should have performed in Italy.
No, yeah, I did.
But I discovered that it is only the UK that has this restriction. Yeah. They do in Germany, they do in France, exactly. They do in the US.
Yeah, that's why I was asking. I thought there was a change in policy.
No, unfortunately not.
Okay.
Well, what is interesting is that if you look at which are the cells that are transduced, it is only the cardiomyocytes. Despite here the promoter is CMV, so it should be expressed in all cells. But it is not expressed in fibroblasts, not expressed in endothelial cells, not expressed in monocyte. Even 10% of the heart is monocytes, but it works only in cardiomyocytes. The vector is very simple, and we believe it is very efficient. But you have to consider that here we injected 6.6 by 10 to the 10 viral particles. Here systemically 2.2 by 10 to the 11. If you go to patients, you inject doses of 10 to the 14 viral genogram per kilogram. And now there have been more than 1,500 patients that have been administered systemically this AV9 vector for spinal muscular atrophy, the one I was telling you before, motor neuron disease. And 1 out of 3 has experienced an adverse event or toxicity. Some patients in other trials have died. Some other patients have a lot of side effects because you inject really an enormous amount of vector. This is a calculation that was made. If you want to target the skeletal muscle fibers, there are approximately 250 million of these fibers, so 2.5 by 10 to the 8. And here, if you inject 10 to the 14 viral particles per kilogram in a patient with 70, you inject 7 by 10 to the 15. So this is 7 orders of magnitude more viral particles than the target cells. So really an enormous amount of vector. And this is not because of the receptors, but it is because of what happens after the receptor. So endocytosis, it has to escape from the endosome, traffic into the nucleus, be imported in the nucleus, has to have single strand to double strand genome conversion. You have to express the genes. So this is an old experiment that we did. This is CHO cells, Chinese hamster ovary cells, poorly permissive to VEGF-P. See only 2 cells, but you treat these cells with hydroxyurea that acts as a helper, and then this is efficiency of transduction, so enormous increase in efficiency of transduction. If you see what happens, you quantify this by flow cytometry, enormous increase in efficiency of transduction. If you look by PCR, how much DNA is inside, it is the same, minus hydroxyurea and plus hydroxyurea. So the vector gets internalized, but then remains trapped in endosomes, or it never reaches the nucleus, is never converted from single strand to double strand. This is another experiment that I love, and we would like with ESA to reproduce also here. This is one that we did when I was still in Pisa. So this is an AV vector that contains 112 repeats of a very simple sequence from Bacterophage lambda, the lac operator, and it is used to infect a cell line, which is the MRC cells, which are poorly permissive to AV. And we engineer these cells to express the lac repressor, which binds to a lac operator, fused to GFP. So basically, there is a background fluorescence in the nucleus, and this is what happens when you give this vector over time. And you see that you don't see the fluorescence that increases or changes, but you see that it accumulates in specific structures in the nucleus, which we call the AV foci. So there is progressive formation of these big AV foci in the nucleus. And we spend really almost 1 year trying to understand what these foci are, until we discovered that they are the same places where the protein of the cellular DNA damage response go. So MBS1, RAD50, MDC1, MR11, the activated form of MBS1, they all co-localize in these AV foci. MRN complex is a complex of 3 proteins. These 3 proteins that are known to recognize single-stranded DNA and DNA breaks and keeps the chromosome together. It's the first line of response to double-stranded DNA, double-stranded single-stranded DNA damage, by which we developed a model by which a cell that is non-permissive has the AV entering, but then it reaches the nucleus, it is blocked by these DNA damage response protein, which blocks single-strand, double-strand conversion. They try to repair this DNA, but also they inactivate. Instead, the permissive cells, this doesn't occur, so the vector becomes from single-strand to double-strand. And we believe that the reason why the cells, like the heart, are not permissive, it is simply because they don't express these DNA damage proteins. In fact, if you inject an AV vector, in this case expressing beta-galactosis, this is another experiment, immediately in a neonatal heart, and you leave it for 7 days, this is efficiency. But if you wait 7 days, this is efficiency. If you wait 21 days, this is the efficiency. And if you look at the levels of these proteins at birth, it is quite high. And then progressively goes down, and 7 days they disappear. Because these cells, cardiomyocytes, don't go into S phase, so they don't need this protein, because these proteins are used for homologous recombination, which require S phase. And the same is for RAD51. RAD51 is the main cellular proteins that recognize the single-stranded DNA, so the virus is single-stranded. It goes into these AAV foci. And this probably is the reason why these vectors are also the vectors that do this high efficiency site-specific targeting, because they are bound by RAD51, which is a protein that is required for homologous recombination. You can deliver this concept also in vivo. This is an AV vector expressed in GFP, injected in the portal vein, where you see this is a level of transduction. But if simultaneously you give an siRNA against RAF50, MR11, or MBS1, you see this level of transduction. Miguel here has performed a high throughput screening, testing all 20,000 human genes, knocking down all 20,000 human genes to see which are the genes that regulate AV transduction, found a series of genes that are very effective, and the top siRNAs against these genes are effective, very effective in this system. This is a basal transduction. This is the same dose of vector after you knock down each of these genes. And Isa is doing the same for the heart. Basically, she can screen microRNA case. She found microRNAs that are very effective to increase the transduction. So this is a low dose of AV vector in cardiomyocytes, and this is the same low dose together with the microRNA. These microRNAs, and this is also in vivo. It is you see it here. I don't know if you can. This is a low dose given systemically, so you see this is the basal transduction of the heart. But in this position, she injected an LNP from gang that expresses, that carries one of these microRNAs. You see that only in the region of the heart where the microRNA is present, she has a massive increase in transduction. So this would mean that with these microRNAs, we could lower significantly the dose and go into subtoxic range. Final slide is this one. Don't think of vectors just to cure diseases, but they are a fantastic tool for investigation, because instead of generating a transgenic mouse, which takes you 1 year and a lot of money, you can develop a vector, inject in vivo, and see the phenotype. For example, if you inject a vector with interleukin-1 beta, you see infiltration of the skeletal muscle with a lot of lymphocytes, same for semaphorin 3A. If you vector for VEGF-A, you see a lot of formation of arterioles. You can play also mixing different vectors and see what they do. This is a very, very interesting game. I stopped here, and I took a lot of time on gang, so gang.
Thank you very much. Mario, do you have the pointers? Oh, thank you. Good afternoon, everyone. My name is Gang Li. I'm the postdoc research fellow working in Mario's lab. And thank you very much for the opportunity to speak to you today about LNPs. And I will be focusing today on the production and application for RNA delivery. So first of all, I want to give you an overview of this, what my presentation is about. So first of all, I will introduce you a little bit background of what stable nucleic acid lipid nanoparticle and their landscape in clinical application. Then I will talk to you a little bit about the formation and the comparison with AAV. Lastly, I will introduce you a lot of application, how we apply this SNAP technology in the case of RNA delivery. I will mainly focus in the heart, because this is being an organ that's a formidable barrier for the LNP to penetrate. And what is stable nucleic acid? So this is was developed in 2001, and the evolution of cationic liposome like lipofectomy for delivery of nucleic acid. It's quite small. It's around 100 nanometers. It's like 1,000 smaller than a human hair. So it's a microscopic particle, barely a low surface charge, and remain intact in circulation for many hours. And in particular, we have 3 FDA-approved products. First of all, called Ampatro for the treatment of polyneuropathy. This is a disease characterized by simultaneous malfunction of multiple nerves in the peripheral. And what happened with this drug is designed to target a gene that encoding amyloid. So in polyneuropathy, the amyloid accumulates excessively in the nerves, which affects which lead to the dysfunction of the nerves. With these drugs, once it's injected intravenously, it goes to the liver and containing. So this drug contains siRNA. It will break down amyloid. So we see a reduction of amyloid. Later on, you see an improvement of this disease. And also we have with the COVID vaccine from Pfizer and Moderna. Without them, I think we wouldn't be here. So this is the mRNA-based therapeutic. So this RNA is encoding with one part of the virus called spike protein. And once we inject this RNA in the form of lipid nanoparticle, so it will train the immune system to recognize this specific protein. And as soon as we encounter later on, this the immune system will recognize and kill the virus. And the other thing I want to touch is what is the composition and how we can adapt into different applications. So it consists of 5 components, including the RNA. So first of all, you have ionizable lipid, which is can be protonated at the pH 4 at acidic environment to electrostatic with the nucleic acid. And then we raise the pH to physiological condition, it will become neutral, so remain intact in the circulation. We also have a structural lipid and cholesterol, which maintain the stability of this biolayer. And also we have pegylated lipid. When we have pegylated lipids, confer the stability in the circulation, so you don't have the problem of drugs coming out. And what is remarkable about this technology is its versatility and highly amenable to different routes of administration. As you see previously, we can give intravenously, we can give intramuscular, we can also give intranasal, intratumor, also sometimes give intracardiac injection. So it's highly versatile in different routes of administration. Oh, sorry. Okay. Oh, sorry.
Sorry to disturb you, but it covers some part
Oh.
of the slide.
Sorry, I'm trying to navigate the mouse. Okay, I'm here.
Oh,
No.
it's okay.
Maybe then. Trying here. Probably let me do like this. No.
Try pressing the arrow down here to make it smaller.
Probably I couldn't find.
No, okay.
Like this?
Okay.
And then, sorry, go slow. I'm sorry, I think it doesn't work, unfortunately. So, okay, we are in this part. So, what I want to talk about here is, regardless of different routes of administration, they are making a similar fraction. So, in here, what we have is a lipid ethanol, RNA in water. If we mix them, so we mix them, and then we will end up with a mixture of ethanol and water. What happens is when we have cationic lipid, which will precipitate out of solution in here, this form this like zigzag pocket you see, and then together with cholesterol structural lipid, we co-encapsulate the RNA. But the problem for this one is the metastable is unstable. So we must mix RNA and lipid in a very rapid manner in the form of milliseconds. And what happens is after you mix so rapidly, you end up with the packaged lipid, maintain the stability of the complex. In the end, you have this biolayer. And the second part is in terms of mode of action. Once endocytosis is into the cell, what you have is this RNA and lipid trapped into endosome. Because of the environment is acidic, so this ionizable lipid will be protonated. And together with the other lipid, this will cause a conformation change inside endosome. And in the end, especially in the case of, let's say, siRNA or mRNA, will erupt the endosome, you see the drug release. For example, mRNA, where the mode of action is a cytoplasma or siRNA. So this will exert the therapeutic effects. And this is kind of the mode action for the SNAPs. And what I want to talk here is the landscape in the clinical development. As you probably already appreciate, we have the SARS-CoV-2 with the Pfizer and Moderna vaccine. We also have the first mRNA drugs approved in the market. You also have a vaccine against influenza in phase 3. And also you have the Zika virus. What's recently quite promising, also quite trendy, is the personalized cancer vaccine. So this will be, so what happens is with this cancer vaccine, so scientists sequence individual patient who has the tumor and to check if they have a specific antigen that's specific for this tumor. Once they identified antigen, they will create mRNA against this antigen, and then this will help the immune system to recognize. Later on, once the immune system recognizes, it will also recognize the tumor which has expressed this specific antigen. Then this is quite trendy at this moment to develop personalized medicine in terms of cancer. And this, I think in the audience, we're probably all aware of AAV as a delivery system. And what I want to point here is a comparison of SNAP versus AAV, so that when you are later on, you want to decide which one you want to pick up for your application, you have both in your mind. So first of all, I would say both of AAV and SNAP are excellent tools for gene transfer. And with the SNAP, you have a unique advantage where it can package nucleic acid of any length. And the SNAP can allow repeated administration. With the virus, we are limited. We can only have like one dose. And the other thing with the SNAPs, you can have a transient transfection. In the case of acute event like myocardial infarction, which we want immediate effects. So at this particular scenario with the SNAP probably is an advantage because once we give the drugs, it finishes what they want to do, stabilize the disease, it quickly eliminates. So we reduce the toxicity. But for the therapy which require long-term, like lifelong protection, AAV, I think is excellent tools. So here is and once we know we have the SNAP in mind, how do we go about doing this? We have spent so much time, a very long time, to set up a facility where we can develop and we come up with the comprehensive steps to facilitate this RNA medicine development. First of all, we can identify what kind of disease target, then we design nucleic acid either to silence, express, or edit. And for mRNA therapeutics, we have we can help with the design of plasmid from and then carry out IVT to get mRNA. Then later on the stage, we can purify with ACTA to get with chromatography to remove the double-strand RNA or unreacted nucleotide. So we get the pure RNA. And then we have two systems at the small scale with just basic vortex. This is particularly suitable for testing in cells, in mice, where we don't require large quantity. Well, we also have excellent tool with a large scale called microfluidic mixing. You have RNA and lipid mixed in a micro tiny chamber, and then in the end you have the lipid nanoparticle. This is kind of microfluidic mixing. It's also the technique they have been using from Pfizer and biotech, where they manufacture at the global scale. And once we have this RNA, we package into lipid nanoparticle, we have quality control to make sure they are the right characteristic. We can check the size, the charge, and the encapsulation. Then once we have this in place, we can so we have the LNP ready to test in cells, or in mice, or in pigs. So if you are interested in accessing with all this equipment, you can feel free to contact me. I have my contact here, so we can help you with these steps. And then we also have access with the lipid toolbox. For example, we have access with the Moderna lipid, Pfizer lipid, and Patrio from siRNA drug. We also have the other list of lipid due to the space I haven't mentioned in here. We also have the structural lipid, different sort of forms. And regarding the testing system to screening the LNP for gene transfer in the heart, we have a large collection of testing model. We can test in primary cell from rats or mouse neonatal cardiomyocyte. We also have ex vivo where we take the tissue from the heart, for example, rat myocardial slices and a human myocardial slice. This is from the heart failure patient. Then we take from the heart, the tissue from them, and we make cardiac slices. They are 300 micrometers thick. And then this is an excellent tool to assess whether the formulation we have can be can work in human samples. Also, in terms of in vivo testing system, we have in mice and pig with healthy mice of myocardial infarction. And we also have, so here is the simple testing method. We test with the EGFP mRNA because it's so simple. We can test 24 hours injection. We can take the heart and check the immunofluorescence to assess the transfection and the cell specificity. So within 1 week, we can test how this LNP system perform in the heart. So it's a really quick assay. And again, if you are interested in using the system, you can contact me. I can make you relevant connection in here. And once we have these excellent tools, then we start this work by testing EGFP mRNA over 30 formulations. We inject them in the heart, 24 hours, we collect them. And the heart is subject to immunofluorescence to check the GFP. So it's quite interesting to know that with Moderna formulation, SM102, this formulation, when you inject in the heart, primarily transfect non-myocytic cells, as you see in here. And then this is the GFP in red. We have another formulation from Pfizer, ALK, and now you can appreciate we have a cardiomyocyte transfection based on the rod shape of the morphology. We also have non-cardiomyocyte transfection, as you see in here. We also test another one. This is the work collaborated with the Hackett Therapeutics. This is the King spin-out company, which works on cardiac regeneration with non-coding RNA using LNPs. With this particular formulation from Hackett, we can see a really high level of cardiomyocyte transfection, as you see in here. Really a nice targeting effect if we inject directly in the heart. Then we test the same formulation in the pig heart. As you can appreciate, this is a big chunk of tissue. So with the pig heart, we inject them directly in the heart. 48 hours, you see in here, we have the nice transfection in a pig myocardium. And this is really encouraging result. Another application I want to show you is on the space of cardiac protection. So we're probably all aware that during heart attack, we lose a lot of cardiac tissues, meaning cardiomyocyte. So what we want, and over time, we will have heart dilation and fibrosis. So what we want is a therapy that can protect the heart immediately after myocardial infarction. And at the same time, when people have a myocardial infarction, sometimes they give the perfusion to limit the infarct size. So with this particular, but the problem with perfusion is that this has a huge oxidative damage in the heart. What we want is to reduce the oxidative damage from the heart with so we take advantage of the liver tropism of LNPs with the circulating cardioprotector called cDNA that has been identified with AV screening previously. So what we have is liver tropism LNP together with the cDNA in the form of mRNA. We give this together intravenously in the mice with the myocardial infarction. Over 1 month period, what you see is in the echocardiography, after 1 month, you see an improvement of heart function. You see the ejection fraction goes up. And also on the cardiac pathology, you see the fibrosis reduction, and we see reduce of scar formation. So this is in the context of cardiac protection. We also, in the space with the cardiac regeneration, we work with the Hackett Therapeutics. We develop microarray that can stimulate cardiac regeneration or force the cardiac cell to re-enter cell cycle. So what we have in here is the LNPs formulation encapsulated with microarray, and we inject them directly in the heart. At the same time, we have a myocardial infarction in the mouse. So in the mice, after 1 month, we check with echocardiography to see the function of the heart. As you can see, we improve the heart function. Also on the histology, we see a reduce of scar formation. So this is in another area in the context of cardiac regeneration. We can use LNP to deliver. And another areas which is really exciting on the gene editing. So we start. So you probably have already aware people have used AAV to deliver gene editing component in the heart, but nobody has managed to demonstrate using LNPs. And I haven't got enough data, but what I want to show you here, we are working on these areas on either CRISPR, base editing, and prime editing. Maybe when we meet next time, I can show you more progress on all of this. And then I want to give you a summary about what I've been introduced to you. So it's the introduction of stable nucleic acid lipid nanoparticle, the formation of SNAP, direct comparison with AAV. And we have an LNP SNAP platform to deliver different nucleic acids in the heart. And then we are open for collaboration with a wide range of cardiovascular research using LNPs. Here's my contact. If you are interested, you can contact me. And then I want to finish thanks to all these amazing people from our lab and the amazing technician team. Without them, I think they are ensuring we run smoothly on the project. And thank you very much for your attention.